Hello, everybody. Welcome to MicroTalks. It looks like we still have some people trickling in, but I'll get started with introductions so that we give our speakers the full time. First off, I'd like to introduce Francesca Scribano as our first speaker. Francesca got her undergraduate degree from Augustana College with a BA in biochemistry, and she's currently a Ph.D. candidate from the lab of Dr. Joseph Hyser. And today she is going to tell us about intracellular calcium signaling in Tulane virus infection. Take it away, Francesca.
Just share my screen first. Can everyone see everything okay?
Yes.
Perfect. Okay. Thank you for the introduction and for the invitation to be here today. So this afternoon, I'll be talking to you guys about my research project, which focuses on calcium signaling and enteric virus infection. And this is of great importance because intestinal pathogens represent a significant global health burden. And every year, diarrheal diseases cause about 1.6 million deaths worldwide, making them the 8th leading cause of death among all age groups and the 5th leading cause of death in children under the age of 5. Importantly, enteric viruses are the leading cause of gastroenteritis worldwide, and these include things like human norovirus and rotavirus. So we can imagine the cycle wherein an infectious disease causes diarrheal symptoms. This leads to physiological stress, which can then feed back and make an individual more susceptible to infection. And this is particularly exacerbated by lack of specific antiviral therapies or vaccines. If, however, we can prevent or treat an infectious disease before we create a pathophysiological state, then we have the chance to break this cycle. So it's critical that we understand the host pathogen interactions that occur during these enteric virus infections. And the 3 viruses that I'll be talking about today are rotavirus, human norovirus, and Tulane virus. And I'll mostly be showing or talking about foundational studies in rotavirus and then detailing how we are able to translate some of these findings to Tulane virus and also human norovirus. So just as a point of comparison for those who may not be familiar, rotavirus is in the family Rheoviridae, while human norovirus and Tulane virus are both in the family Calicivirus. Rotavirus has a double-stranded RNA genome, while human norovirus and Tulane virus are positive sense single-stranded RNA viruses. Rotavirus is an important human pathogen, particularly in pediatric populations. Every year it causes almost 130,000 deaths and about 258 million episodes of diarrhea. Human norovirus is also an important human pathogen across all age groups. And so every year worldwide, it causes over 200,000 deaths and about 685 million episodes of diarrhea. Tulane virus was isolated from rhesus macaques in 2008. And while it is not a human pathogen, it has often been recognized as an important model system for human norovirus. Human norovirus has historically been a challenging pathogen to study, and this is due largely to the fact that it cannot be cultivated or propagated in conventional cell culture systems. And so to address this problem, our lab uses Tulane virus, which is a very closely related rhesus enteric calicivirus. And Tulane virus is such a useful surrogate because it allows us to conduct mechanistic studies, which we can then ultimately confirm in the context of human norovirus infection. And Tulane virus is such a useful starting point because it shares many similarities with human norovirus, including its genome organization, its tropism for intestinal epithelial cells, and its acute diarrheal disease course. So now that we have the tools and the models to study important pathogens like human norovirus, we can begin to ask some interesting questions. And I'm specifically interested in the dysregulation of cellular calcium that occurs during infection. We know that many viruses are able to hijack host cell signaling dynamics, and one of the more direct ways that this is achieved is through virus-encoded ion channels or viroporins. So in this figure, I've just boxed out virus families that contain at least one virus with known viral ion channel activity, just to emphasize how ubiquitous this actually is. So as I mentioned, these viral ion channels or viroporins can localize to internal stores of ions such as calcium and mediate their conductance. And if we look specifically at enteric viruses, we can find many examples of either ER or Golgi localized viroporins that specifically target calcium. And one known example of this is rotavirus NSP4. So Ali Surtek was a former graduate student in the lab, and she was able to begin to characterize the dysregulation of cellular calcium that occurs during Tulane virus infection. And so to do this, she used cell lines that were engineered to stably express a green cytosolic calcium indicator. So we'll watch these time-lapse videos, and anytime we see an increase in green fluorescence, that means an increase in cytosolic calcium concentrations. So we have mock infected cells on the left and Tulane virus infected cells on the right. And when we play the video, we can almost immediately appreciate that the calcium signaling in the Tulane virus infected cells is much more dynamic than in the mock infected cells. And this is actually infectious dose dependent. So if we compare to mock or irradiated Tulane virus, when we infect our cells with increasing multiplicities of infection, we see significant increases in the number of calcium spikes that occur, suggesting that during Tulane virus infection, this increases cytosolic calcium concentrations. Ali then went on to show that the onset of these calcium signaling dynamics seems to coincide with viral protein expression. So here we're tracking expression of the non-structural protein NS12 in red. And we can see that the onset of these calcium spikes or these calcium signals really coincides with NS12 expression. And this is the case for other known calcium conducting viroporins, like I mentioned for rotavirus NSP4. Ali also showed that transfection of the non-structural protein NS12 alone was sufficient to mediate the significant increases in the number of calcium spikes that we were seeing, again, consistent with other known calcium conducting viroporins. So this suggested that Tulane virus infection can dysregulate calcium signaling within the infected cell, and this is likely through viroporn activity of the NS12 protein. As I mentioned, one of the utilities of Tulane virus infection is that we can try to translate our findings to human norovirus. And as I mentioned, human norovirus can infect conventional cell lines, but we can transfect in individual proteins from human norovirus. And here what we're seeing is, again, tracking the calcium signals that occur with transfection of the non-structural protein NS12 from human norovirus G23. And again, seeing that the onset of these signals really coincides with protein expression. And again, the NS12 protein from human norovirus alone is sufficient to mediate significant increases in the number of calcium spikes within the transfected cell. And this is to a similar degree as Tulane virus NS12, suggesting that the NS12 protein from human norovirus may additionally demonstrate viroporin-like activity. So just to demonstrate the data that I have alluded to or shown you already, and then to kind of forecast where I'll be going with the rest of the talk, I just wanted to summarize that it's been long established in the literature that rotavirus infection causes significant increases in cytosolic calcium concentrations. And I've shown you data to suggest that the same is true with Tulane virus infection and human norovirus transfection. Again, it has been established that rotavirus NSP4 is an ER localized calcium conducting viroporin. And the same is true for the NS12 protein of Tulane virus, and likely also the case for human norovirus NS12. And kind of the last calcium signaling phenotype that I'll talk about for the rest of my talk today is called intercellular calcium waves. And these were again first identified during rotavirus infection. And we are interested to see whether or not this might also translate to these other viruses. And this was particularly given the similarities that we had already noted between cytosolic calcium concentrations and viroporn activity. So just to demonstrate phenotypically what we mean when we refer to intercellular calcium waves, here we see cells again stably expressing a cytosolic calcium indicator. So we'll see cytosolic calcium in green, and then our rotavirus infected cells in this magenta color. So particularly during rotavirus infection, we can appreciate that there are these calcium signals that occur not only in the infected cell, but also in the neighboring uninfected cells. And this occurs in this very coordinated intercellular calcium wave phenotype pattern. And then we can actually on paracrine purinergic signaling. So ADP is based from the rotavirus infected cell, and it can bind the ADP receptor P2Y1 on neighboring uninfected cells. And it's this binding of ADP to the P2Y1 receptor that elicits the cytosolic calcium flux in the neighboring uninfected cells. So the two main questions that I'll address for the rest of today are, one, can Tulane virus also propagate intracellular calcium waves? And are these waves also dependent on paracrine purinergic signaling? So to go after the first question, I just wanted to go through in a little bit more detail what my approach was for generating the time-lapse videos that I'll show in the coming slides. So again, using cell lines that stably express the cytosolic calcium indicator, we can then infect with Tulane virus. And then we can capture still images throughout the course of infection, stitch these individual images together to generate our time-lapse movie. And then after we have the data for our time-lapse, we can actually fix our monolayers and stain, antibody stain for Tulane virus antigen, take a still image of our antibody stained cells, and then overlay this still image with our time-lapse video. And this is to be able to distinguish infected from uninfected cells in our monolayer. And then once we do that, we can quantify the number of calcium spikes per cell that occur. And then one important point is that both rotavirus and Tulane virus can infect two different cell types. The first are MA104 cells. So this is a monkey kidney cell line that was derived from African green monkeys. And the second is an LLC MK2 cell line. This is also a kidney cell line, but was derived from rhesus macaques. And so all of the rotavirus infection data that I've shown you to this point was done in MA104 cells, and all of the Tulane virus studies that I have shown to this point were done in LLC MK2 cells. So I wanted to test whether or not if infected MA104 cells, I could see this intercellular calcium wave. So we have mock and pretty zoom in on one particular infected cell, we can appreciate again these signals occur not only in infected cells, but also in the proximal uninfected cells. Okay, so we can kind of qualitatively appreciate this intercellular calcium wave phenotype with Tulane virus infected MA104 cells by watching the time-lapse videos, but we can also look at this quantitatively by comparing the number of calcium spikes per cell that occur in mock infected cells, in Tulane virus infected cells, and then in the cells that are neighboring infected cells in our TV infected monolayers. And we can see that in MA104 cells, there are significantly more calcium spikes in the neighboring cells compared to mock. And so this is consistent with our Tulane virus infection of MA104 cells causing intercellular calcium waves. Interestingly, as I mentioned, all of the previous Tulane virus infection data that I had shown you was done in LLC MK2 cells. So now that I had this way of antibody staining and tracking Tulane virus infected cells, I wanted to see if I could see waves in my LLC MK2 cells. So again, we have mock infected cells on the left and Tulane virus infected cells on the right. And we see plenty of Tulane virus infected cells shown here in red in this monolayer. And we still see lots of sporadic calcium signals occurring within an infected cell, but there are none of these coordinated signals that spread outwards to the neighbors. And again, looking at this quantitatively, there's no significant difference in the number of calcium spikes between my mock and my neighboring cells. Consistent with the idea that Tulane virus infection of LLC MK2 cells does not propagate intercellular calcium waves. So it's really interesting to see this difference between cell lines. And so we wanted to actually go back to rotavirus kind of as our positive control and see if we would also see the same differences between cell lines. So we went back and we infected MA104 cells with Tulane virus, we infected MA104 cells with rotavirus. We have mock infected on the left and rotavirus infected on the right. We'll see our rotavirus infected cells come up in this red color here, and we pretty immediately see these very coordinated intercellular calcium waves that occur. Again, when we look at the quantification, significantly more calcium spikes in the neighboring cells compared to mock. supporting all the data that we had seen previously that rotavirus infection of MA104 cells can elicit intercellular calcium waves. And what was new was going back to infect our LLC MK2 cells now with rotavirus. So again, mock on the left, rotavirus infected on the right. You'll see our rotavirus infected cells come up in this red color here. Again, we see lots of kind of sporadic calcium signals, and the infected cells themselves are signaling a lot, but we see no coordinated signals that spread outwards to the neighboring cells. So rotavirus infection of LLC MK2 cells also does not elicit waves. So this pointed to this really important difference in the cell lines. So both rotavirus and Tulane virus propagate waves in MA104 cells, but neither can propagate intercellular calcium waves in LLC MK2 cells. So we were interested to see what the difference was between these two cell types. And one critical thing that we were able to note is that LLC MK2 cells do not express the P2Y1 ADP receptor. And this was kind of the first line of evidence that suggested that purinergic signaling may also be important for Tulane virus-induced waves. And this also set up a nice way, or a nice genetic approach, to test the necessity and sufficiency of ADP P2Y1 signaling for waves. So you could either knock out P2Y1 in the MA104 cells and see if we lose the wave phenotype, or introduce P2Y1 into our LLC MK2 cells and see if we can rescue our wave phenotype. And these genetic approaches are ongoing. But what I have done so far to analyze whether or not Tulane virus-induced waves depend on paracrine purinergic signaling is to use pharmacological approaches. So here in MA104 cells, mock on the far left, our DMSO vehicle control in our second panel. And then we use two different drugs. The first is a pyrase, which degrades extracellular ADP. And the last is BPTU, which blocks the P2Y1 purinergic receptor. And in these videos, we can see that in our DMSO vehicle control, we see our nice coordinated intercellular calcium waves. But this phenotype is significantly attenuated in our A-pyrase and BPTU treated cells, suggesting that purinergic signaling may be a conserved mechanism of virus-induced waves. So the big remaining question in the future directions is kind of aimed at why do we care about these intercellular calcium waves in the first place, and what can they tell us about virus-host interactions? And interestingly, we were able to see that the intercellular calcium wave has kind of seemingly contradictory both proviral and pro-host effects. So for the proviral effects, if we look at virus replication, first in rotavirus, we know that if we degrade extracellular ADP or block the P2Y1 receptor, we see a significant reduction in rotavirus yield, and the same trend is seen with rotavirus. On the other hand, if we look at pro-host effects in the production of cytokines and different types of inflammatory mediators, we know that in rotavirus infection, certain cytokines such as IL-1 alpha are initially upregulated during infection. But again, if we block the ADP P2Y1 signaling, then we see significant reductions in these cytokines. And while I haven't done the pyrase and BPTU treatments for Tulane virus infection yet, the same type of cytokines that are dependent on purinergic signaling are also upregulated in Tulane virus. I'm showing data for IL-1 alpha here, but this is also the case for other inflammatory mediators like COX-2 and INOS. So the take-home messages from my talk today have been that calcium dysregulation is a conserved feature of many virus infections and many enteric virus infections. That Tulane virus is an important model system to understand how caliciviruses can dysregulate calcium signaling. That calcium dysregulation, particularly as it extends to neighboring uninfected cells, is also conserved across multiple viruses and depends on host cell purinergic signaling. And finally, this is important because the ADP P2Y1 signaling axis is a potential broad spectrum and host-directed antiviral and therapeutic target. So with that, I would like to acknowledge all of the wonderful members of my lab, and I would be happy to take any questions.
Thanks, Francesca. While we're waiting for questions to come in, I'll ask a question. So what cell types express P2Y1 in humans? Is it just enterocytes or is it a variety?
Yeah, so there are actually all different types of cells that express P2Y1. You see them in intestinal epithelial cells. You see them expressed on cells like macrophages. So P2Y1 is actually very broad. So the rest of P2Y1 is expressed throughout the human body.
I have a question, Francesca. So you mentioned at the end of your talk that it would for humans. So I was wondering if it's both ways, if I understand it. So it reduces viral replication, but it also reduces the host response. So how do you see using it as a therapeutic?
Right, so that's a great question. So we don't know yet whether or not the reduction in cytokine responses that we're seeing are kind of secondary to the differences in virus yield. You know, if you have a lower level of virus infection, then you're not going to mount as strong of an immune response to that virus infection. We also see that some of the cytokines that are upregulated are not necessarily directly antiviral. Like IL-1 alpha I showed data for is not usually thought of as directly antiviral, but it is involved in kind of restitution and barrier repair following infection. So it's possible that if we kind of target ADP P2Y1, you know, we would still be able to attenuate virus infection and hopefully also decrease. If they're not direct viral, hopefully that won't have a competing effect.
Great, thank you. Very nice talk.
Thank you.
Any other questions? All right. So thank you, Francesca. Next, I'd like to introduce our second speaker, also hailing from the Hyser lab. So Kristen Engevik got her BS from Baylor University in California and then went on to get her Ph.D. in systems biology and physiology from the University of Cincinnati in the lab of Marshall Montrose, studying the mechanisms of TFF2-mediated gastric restitution. She currently is a postdoctoral fellow in the Hyser lab, where she has postdoctoral fellowships from both the American Physiological Society and the NIH National Institute of Diabetes and Digestive and Kidney Diseases. So today, Kristen is going to tell us about viral exploitation, how rotavirus uses host signaling for pathogenesis. Take it away, Kristen.
Great, thank you for that introduction and this opportunity to share our work from the Hyser lab. It's going to be very Hyser-centric today, and I'm going to be building off a little bit of what Fran has already shared, just transitioning into rotavirus, not Tulane virus. So, as already mentioned, diarrheal diseases still remain a very serious problem worldwide and are among the leading causes of death. And this is especially true within children, where diarrheal diseases can cause about 2 billion cases and result in, especially within children, in 500,000 deaths each year. And among some of the key culprits causing diarrheal disease are enteric viruses, which make up for about 40% of the diarrhea cases worldwide. And specifically, the enteric virus, rotavirus, which Fran touched on a little earlier. Now, those who are afflicted with rotavirus infection, unfortunately, the only suitable treatment for children is oral rehydration solution. And while it is effective at replenishing fluids, it has no change in diarrhea severity or duration. And often there is poor compliance. And ultimately, this highlights the need for us to develop suitable pediatric antidiarrheal drugs. In order for us to develop such things, we need to have a better understanding behind the pathophysiology of diarrheal infections. And we chose to focus first on rotavirus, as it is one of the key causes of diarrhea cases. And it life-threatening diarrhea causes again, protection of vaccines against rotavirus have proved to be very effective in high-income nations such as the US. These vaccines have proved to be far less efficacious and effective in lower-income nations where rotavirus infection is still most rampant and detrimental. And this highlights the need for us to have a better understanding behind the pathogenesis rotavirus effects. This is a very localized infection across uninfected all time. Long been with rotavirus touch cell pathology, including chloride secretion from the enterocytes, as well as stimulating serotonin release from enterochromaffin cells. And this exacerbates fluid secretion by activating the enteric nervous system. In fact, this activation of the enteric nervous system has been considered a critical part of rotavirus diarrhea, such that treatment with serotonin receptor antagonists have been a proposed therapeutic to treat rotavirus disease. Thus, looking at paracrine signaling can be likely responsible for a pathophysiology that's associated with infection and could be a more viable target to alleviate rotavirus disease without having large systemic effects. So, as Fran eloquently showed earlier in today's talk, one hallmark of rotavirus infection is that rotavirus increases calcium within the infected cells, which is important for viral replication, but this has also ultimately been shown to contribute to diarrhea. And this allows us to use calcium as a tool to look further into that paracrine signal and also looking at rotavirus infection over time. And so similar to what you've seen earlier, we utilize live imaging using those monkey kidney cells, MA104, which express the calcium indicator shown here in green. And what you can appreciate in these videos is that calcium is increasing not only in the infected cells, which are indicated by the magenta, but then you also see these wave-like projections that Fran focused on that are propagating from the infected to uninfected cells. So, and this was one of our initial observations that kind of suggested that these calcium waves are a result of the paracrine signal that's released from these rotavirus infected cells. For those observed waves, former Ph.D. student, Dr. Alex Chang-Graham, did a number of experiments looking at potential candidates, which included the rotavirus enterotoxin, ENSP4, gap junctions, prostaglandin, nitric oxide, and the purines, ATP and ADP. And spoiler, as you probably have already gathered based on Fran's talk, what we were able to find is that these calcium waves occurring during infection rely solely upon purinergic signaling of ATP and ADP. And just to repeat what Fran had shown, when you hydrolyze the availability of ADP and ATP, you definitely dampen those calcium waves that occurred during infection. So just as a little background behind purinergic signaling, and then going further into that P2Y1 receptor, which we're interested in, purinergic signaling purine receptor upon as a the G protein covers the act of ness and letter to and Curians have number of test functions, including mobility, appreciation, and absorption. And are to right. So, and what I'm able to see in human is elicit the pure decision then elicits within calcium in and while the great reduction understand receptors found along the GI tract are P2X4, P2Y1, and P2Y2. And responses by nature and nutrition into an in vivo model in order to elucidate whether or not this P2Y1 is only contributing to diarrhea. Our first look the and while entering rotavirus, we see 7 the age, and then of course of that infection so we assess P2Y1 is involved in this mild model of disease. And so look at scores on a score for mice where normal stool 1 and diarrhea known out of age and then monitor them over the Hyser state when we're looking at the scores. RTU treated lower and what you see diarrhea score compared to the PBS groups. Furthermore, when you look into using a more competitive P2Y1 inhibitor known as MRS-2500, we also see a similar trend where we have a significant decrease in the diarrhea score within our MRS treated groups in green compared to the PBS in red. And this ultimately pointed to the fact that P2Y1 inhibition can at least ameliorate the mild disease that is being caused by our heterologous infection. And so this led us to wondering whether or not inhibiting P2Y1 can alleviate a severe model of rotavirus infection, which would be gavaging our mice or infecting our mice with a mouse rotavirus. And so again, taking our neonate pups at age day 5 to 7, we gavage them and then we monitor them up to 5 days. And similar to what I showed you in the last slide is we then start to treat them with either our vehicle or our P2Y1 inhibitor. And then we collect them at day 3 and 5 post-infection. And we assess daily their diarrhea and viral stool shedding. And then post-euthanasia, we collect the small intestine to assess host responses. So first looking into diarrhea and whether or not this inhibition has a, if P2Y1 inhibition has an effect on the diarrhea score within our pups, we take that diarrhea score where again, anything ranked over a 2 is considered diarrhea. And what we observe is in both groups, we have peak diarrhea occurring at day 2 to 3. And starting at day 4 post-infection, we see that there's a significant decrease in our diarrhea score within our MRS treated groups shown here in red. Furthermore, when we look at the diarrhea incidence, where we're taking the number of pups with diarrhea over the total pups that stool for each day, we also see this similar trend starting at day 4 post-infection, where we have a significant decrease in that diarrhea incidence in the MRS treated groups. Now, as we see that there is a key difference in our P2Y1 inhibited mice and alleviating rotavirus diarrhea, we wanted to see if this also had an effect in viral shedding in the stool. And so what we did was prior to treatment each day, we collect the stool and process to look at rotavirus gene expression. And what you can appreciate where we have at day one post-infection, both groups are together as neither have been given any treatment, able to detect viral gene expression in both groups. And at day 2 and 3 post-infection, we see no difference we see no significant difference between either treatment groups. However, starting at day 4 post-infection, we do see that there's a significant decrease within our MRS treated groups compared to the rotavirus or compared to the PBS treated pups. And we think this may suggest that this pure P2Y1 signaling may be having some effect on the rotavirus replication. So as we see that there's a difference within the viral shedding within the stool, we also wanted to see if there's any difference in the number of infected cells within our pups. And so what you see here are some of our immunofluorescence looking at rotavirus, which is stained in the magenta. And we have our PBS treated groups at the top row and our MRS treated group at the bottom at day 3 and 5. And we do see that you can detect rotavirus infection in both groups. However, when we count the number of infected cells within our images that we've collected, we do see that at day 3 post-infection, there's a significant decrease in the number of infected cells within our MRS treated group compared to the PBS. And at least what this is suggesting so far is that this P2Y1 inhibition is resolving a rotavirus infection much earlier than those that are given PBS alone. Now, as we see a difference in diarrhea and viral stool shedding, we also wanted to look further into the role of P2Y1 within the epithelial cells. And as Fred mentioned before, P2Y1 is expressed throughout the intestine and in a number of different cells. So we wanted to first focus on some key contributors, which are which is chloride secretion from the enterocytes, as well as serotonin release from the enterochromaffin cells. So just as a general reminder of enterocytes, they're primarily responsible for digestion, absorption, as well as the innate immune responses throughout the epithelium. And especially during diarrhea and secretory diarrhea, enterocytes do contribute to fluid loss via chloride secretion. And so we want to assess in vivo if through this purinergic signaling with P2Y1, rotavirus is able to elicit increased chloride, as we've seen within our intestinal enteroids. And so in order to look into this question of whether or not blocking our P2Y1 signal could decrease luminal chloride, we take the entire small intestine and we flush it with water and collect the luminal contents. And then with those luminal contents, we form a chloride colorimetric assay. And so what we see here, which is day 3 post-infection, using those luminal flushes in our PBS treated rotavirus infected pups shown in red, we do see that there's a significant increase in the luminal chloride content compared to an age-matched uninfected pup. However, this increase is significantly dampened in our MRS treated pups. Ultimately, so far at least, suggesting that this P2Y1 signaling is required for rotavirus-induced chloride secretion. Now, a key contributor to rotavirus diarrhea that I mentioned earlier is serotonin release from the enterochromaffin cells. And enterochromaffin cells play a very crucial role in the regulation of GI motility and secretion. And so we want to look into whether or not this P2Y1 receptor could be a viable target for that to prevent serotonin release, and whether or not the serotonin release is dependent upon P2Y1 signaling. So to look into serotonin release, we took again those luminal flushes and we performed a serotonin ELISA. And what our data suggests or what we see so far with this, looking at the luminal serotonin content, we do see that starting at day 5 post-infection, we have a significant decrease in our serotonin content in the MRS treated group compared to the PBS. And we also see a similar thing occurring at day 7 post-infection, which suggests that this purinergic signaling plays an important role through P2Y1 for the release of serotonin during rotavirus infection. So to kind of reiterate what Fran so eloquently talked about earlier, I hope what the main takeaway for today is understanding that this purinergic signaling, and especially in the context with P2Y1 in the context of rotavirus infection, is a key pathway in severity and duration of this diarrhea. And that the inhibition of P2Y1, we can decrease that diarrhea severity as well as duration. We can decrease viral, or we can increase rotavirus clearance and viral shedding, and we can also decrease the chloride and serotonin levels that ultimately contribute to diarrhea. And we think that this P2Y1 receptor could be a viable target and a potential therapeutic to prevent diarrhea, or at least alleviate diarrhea overall. And so in closing, I'd like to thank everyone for their attention, as well as the members of the Hyser lab, especially our former member, Dr. Alex Chang Graham, who was the one to identify that this paracrine signal is purinergic in nature. I'd also like to thank our collaborators and our funding sources. And I'm happy to take any questions.
Any questions?
I have a question. Can you hear me?
Yes.
Okay, I have a question about whether or not I'm getting feedback. So let me sort this out before I ask my question. Maybe somebody can go ahead.
Okay, you can also type it in the chat if you'd like. If that would make it easier. Oh, that's a great question, Anton. So I guess I'll read the question first, and in case someone's not looking at that. So Antoine Opukun, I'm sorry if I'm not pronouncing that right, but he says, I can't help but to wonder if P2Y1 inhibition figures into irritable bowel and inflammatory bowel disease as well. I'm not sure. That's a really interesting question, and definitely not something I have looked into. I think it's probably, it's the difficulty with most diseases, especially those within the gut, is that they're usually multifactorial. And I know that there's a lot of different factors that contribute to both IBS and IBD. It may play some role, but I'm not sure exactly if it would be a key role. I don't know if there's any deficiencies or differences in those who are suffering from IBS and IBD compared to a normal healthy patient. But that is a very interesting point. Okay, so Heidi, to answer your question is, so is MRS compound being used for any kind of treatments now? So this, so last I checked, the MRS 2500, that drug is being used for a clinical trial as an antithrombotic. We primarily picked this inhibitor primarily due to the solubility of it, because it's soluble in PBS, making it easier for us to gavage, whereas our previous compounds that we've used, which is BPTU, it's dissolved in DMSO, but for us to give it to our mice, we have to put it in corn oil. So but yeah, it is being used at least for that. I don't know entirely whether it'd be suitable for pediatric use, but yep. Any other questions? Oh, thank you, Heidi. Okay.
Okay.
Hi, Vic. You're on. So, Vic, have you seen or measured any other hormones like melatonin or secretin? Unfortunately, I have not. That would be, it would be interesting to see if there's anything else that's being increased in hormone-wise with during this infection, but I have not looked into that. That's a little bit out of my realm, but that would be interesting to look into. Yeah, thanks, Vic. Oh, I guess I can stop sharing my screen.
All right. Thank you to both of our speakers. We'll be back again with MicroTalks in November, and we'll see you then.
All right, thanks, Penny.